we're going to talk a little bit about clustering. This is going to be very application-oriented, so sort of how to apply clustering and a little bit of background of how the underlying pieces of a clustering algorithm contribute to what you find. So, just to fill a little bit of time and also to show where I'm coming from, this is the NIH campus. This building is called Building 10. They're all numbers. There's no names, which makes it actually kind of fun to find your way around. This building, the back part of this, at the time was state-of-the-art. It was built in the 1950s, and there's propaganda material from the time that every room had a television. But it was also the largest brick building in the world. So this is a city. This building in and of it is a city in and of itself. And then there on campus there are about 70 other buildings. And if you guys have ever been to a government facility, you'll know what it's like. But for me, going to work every day, it's like entering Fort Knox. There's a 12-foot fence around the whole place. So when we're looking at genomic data, I think of these things as views of a genome. In this case, we're talking about views of a cancer genome. I'm talking about views of a cancer genome, but you guys are going to be viewing your own data. And really what we're looking for are ways of distinguishing two different sets of information, trying to find patterns in what can be, in this case, actually pretty simple data. But in other cases, are going to be very complicated data. And because of the technologies that we have, we can measure really everything at every level of the central dogma, right? We can measure DNA replication, transcription, processing, transport, protein translation, post-translational modifications. We can measure all of those things, and typically, you will measure many of them. And so we often need ways to take these various kinds of data, one data set at a time, and make sense of it itself, and then also have ways of visualizing data sets where we can tag features with other pieces of information from other data sets. So clustering helps us. It's a tool that we can use to help us do that. So just to make it really clear, we have gene expression, gene copy number, chromatin structure and function, sequence variation, DNA methylation, transcriptional regulation. You guys are going to talk about many of these things here, right? These all lead to a phenotype. And it's really when we put all the pieces together that we can begin to understand the phenotype. And of course, if people are working with human data, we have on top of the data that we're measuring, and Naomi just gave some great examples, we have to look at patient and population characteristics as well. Otherwise, we can make wrong assumptions. And again, clustering allows us to, both in an unsupervised way, without telling the computer what we think is going on, and sometimes in a supervised way. We know what's going on. We want to see if the data match our assumptions. Clustering can help us with those things. This is just a little picture of our lab. This is 3 out of about 6 or 7 of our Illumina sequencers. This plus this, which is the computer cluster that a couple of us built to deal with our sequencers, equals this, right? And a lot of people feel that way. You know, if you have data, especially if it's really expensive data, you can make this. This is easy, right? You just take your data, you run it through some computers, do some hierarchical clustering, you'll get your Nature paper. Well, what is clustering? I mean, clustering, so when I joined my lab nearly a decade ago, they were still at a point very early in microarrays. But they had discovered one thing, which was how to look at the data that they got out. Their first experiment, I think, had about 1,000 probes on the array, so 1,000 genes. And that, when they did 8 samples, they were looking at different kinds of sarcomas. So they got back essentially an Excel spreadsheet with 8 columns and 1,000 rows. And you guys are thinking, that's really not much data. But this was more gene expression data than had probably been produced in the entire history of mankind. And they began to think about how do we look at those data? How do we think about them? And they thought about it sort of in the same way that this fish key approaches data analysis. They thought about the genes as different features of their samples, in the same way that the shape, the color, the markings are different features of fish. So it's a taxonomy problem. It's a categorization problem. So again, fish, they have their characteristics. Gene expression has a different set of characteristics. This is the usual samples in columns, genes in rows. I challenge any of you to find any structure in that data by looking at it. But if we take the data now and look at, this case, we're looking at 2 samples at once, we can begin to think about how related or unrelated the samples are. So you guys can look at this now, having looked at some gene expression data in the different labs, and you can say that, you know, yes, they're roughly correlated. That is, a lot of the data is along the identity line, but there's also a lot of spread, right? So if we had a way of capturing this, but for all the samples simultaneously, then we could begin to compare the samples to each other. We could measure the differences between samples. So this is how you do it for one sample. Again, you can do it for genes. That's what I was or for samples in columns. Or we can do this in rows as well, right? We can make the same scatter plot for 2 genes across samples. So now we've got sort of a framework for thinking about this. We've got vectors, and we're asking how different are pairs of vectors from each other. And if we do this for all possible pairs, then we can begin to make some sense of the data. So why do we cluster data? Well, I've given you some background. Humans are really not good at looking at large spreadsheets of numbers. We like to find patterns in data. So when we get data back, if we don't find patterns in it, we don't find it very interesting. We don't find it very useful. These patterns help us to define and validate our hypotheses. And notice, though, the hypothesis part here is really important. That scatter plot that I showed you is meaningless in and of itself. We have to attach some biological meaning to it. We need further summaries of the data that are meaningful to drive discovery from large data sets. And finally, clustering, predates microarrays and sequencing by decades. It's one of the well-known techniques for successful applications on very, very large data sets. So millions or billions of data points. Motivations, data visualization, understanding high-level characteristics, generating hypotheses based on data relationships. We can graphically associate covariates with data. So this is getting back to my little circle plot, or my little circle slide. We really need, once we have these pictures of the data, we want to attach meaning to them. We have to attach meaning to them. We'll see that in a minute. In applications, we can identify potential subgroups, groups or subgroups of samples or genes. We can identify outlier samples. We can find batch effects, or hypothesized batch effects. We can determine the strength of a biological signal. So if you do a hierarchical clustering and you see no association with your phenotype of interest, you might have a little bit of trouble. You might have to use statistical techniques that are very sensitive. Whereas if you take all your genes and you see differences, you see that your samples cluster nicely into 2 groups, you may not need much statistical power, or you might have lots of statistical power. So measuring clustering effectiveness, there are probably actually journals devoted to doing this, but at the high level, we're really interested in maximizing the between cluster distances and minimizing the intra-cluster distances. So we want things that are similar to be very close to each other in our clusters, and things that are relatively different to be very far away from each other. So that's something that we can measure within a clustering algorithm. We can also ask how stable is the cluster? If we do the clustering again, but we change things just slightly, do we get the same result? So if we consider just subsets of the data, subsets of the genes, or if we're clustering genes, we consider subsets of the samples, do we get the same clustering when we apply the algorithm again? We add a little noise to the data. How much does that change things? If adding just a teeny bit of noise significantly alters our clusters, then our clustering might not be terribly biologically relevant. We can, and Nomi talked about this yesterday, we can bootstrap on the data. We can take subsamples of the data and do our clustering over and over and over again. And actually, there are people who do just this, and they use the output of the clustering of these multiple types of, or these multiple rounds of clustering as a new distance metric, a new way of measuring differences between samples. And then finally, as you're going to see, there are literally dozens, or actually probably hundreds of clustering algorithms, particularly when you take into account different parameters. So how much does your clustering vary when you change the algorithm or the parameters? First thing I'm going to spend most of my time talking about hierarchical clustering. Are there any questions, though, up to this point? Most of this is sort of just background. So there's really two components to hierarchical clustering, and it's nice to think of them separately, although with computer packages, it's kind of easy to have them, especially computer packages that allow you to point and click. It's easy to sort of forget that these two pieces are important independently of each other. There's the distance metric. Remember I showed you the scatter plot. So on that scatter plot, what comes to mind typically when people see scatter plots is thinking about a distance metric or a similarity metric or correlation. So one minus correlation would be one distance metric. We can also have other distance metrics, and I'll go through those in a bit. And then once we have our distance metrics, our matrix of distances between samples, then we need some way of attaching samples to each other or genes to each other. And so that little piece of hierarchical clustering is called the linkage. How do we attach clusters, first samples or genes to each other, and then once we have clusters of samples and genes, how do we then make the next cluster? There are many different implementations of hierarchical clustering. By many, again, I'm talking dozens or hundreds. Hierarchical clustering is reasonably fast, particularly the clustering part of things. The distance metric can take some time to calculate. It just depends on the distance metric you're using. Another cool thing about hierarchical clustering is that the branch lengths, when we get a cluster back, have some interpretation with regard to hypotheses. Hierarchical clustering falls into sort of two broad categories. Agglomerative, which is beginning with each sample or gene as an individual cluster. So our set S starts with all the elements in our data set, one each. We merge them until S, the set of existing clusters in our clustering approach, finally reaches one, when we have all pieces in one cluster. And this is the most common approach. So when people talk about hierarchical clustering, they're typically talking about agglomerative, bottom-up clustering. Then there's also divisive clustering, which is basically taking the set of data, starting with it as one cluster, and then dividing that into 2 clusters, 3 clusters, until you get to all the possible samples being in their own cluster. So the algorithm is described here in sort of high-level terminology, I guess. The input is this pairwise distance matrix that we were talking about. Again, the distance matrix, the distance metric we'll go into. But that's the input to the clustering algorithm. Then we place each instance into its own cluster, and we create the list of clusters, which initially starts with everybody being in its own cluster. Then we compute some merging cost function, which I'm going to call linkage, between every pair of elements in the list until, and the goal at step 2 here is to merge the 2 closest clusters by whatever linkage method we're using into a single cluster. So the list gets reduced. The list of potential samples that we need, or potential clusters that we need to merge, gets reduced by 1. Then we remove the 2 merged clusters, and so we don't have to deal with them again. Then we merge these things to create a new internal node. So we're creating, again, we're building up the tree. And finally, we go back to step 2. Okay, so a lot of words to describe this process of taking things that are separate and merging them together. So hierarchical clustering in sort of graphical approach, we're going to start with all the genes. We're going to merge the genes that are closest together by our linkage method, or our linkage approach. We're going to keep merging. At some point, we might actually find that this cluster and this gene are closer than any of the other individual genes. So that happens next. And we keep going, merging clusters until we have all of the clusters, or all of the original genes part of the cluster. Yes?
So, using this example, if you were to merge 4 and 5,
Mm-hmm.
and then you were going to merge gene 4, 5, 3,
Mm-hmm.
are you looking at some kind of average of 4 and 5, or?
Yes. So that you're asking a great question. So the question you're really asking is, what is this linkage thing? How are we figuring out how to connect different clusters? Actually, let me jump down here. And just to answer your question as you asked it. So once we have these different clusters, we have some different approaches. Single linkage clustering. We take the distance between one cluster and another cluster to be equal to the shortest distance from any member to any member in the other cluster. So if we have, so in the case of individual genes, that's easy. It's just the distance between the 2 individual genes. But as soon as we have clusters of genes, then all we're doing, this is happening in, if we have 20 samples, this is happening in 20-dimensional space. We're asking, of these 2 clusters, what's the closest point between those 2 clusters? And we're going to do that for all the possible clusters, and we're going to merge the cluster that has the shortest distance. Does that make sense to everybody? Okay. So that the difference between that and the next one, complete linkage, is that the distance between one cluster and another cluster we're going to take to be the greatest distance between any member of one cluster and a member of another cluster. So single linkage, we take the closest point. Complete linkage, we take the furthest point. Otherwise, same thing. Average linkage, this is exactly what you're proposing, which is to take the average distance between all the points in one cluster and points in another cluster. Okay? I'll come back and we'll look at examples of how these things differ and why you might, I won't say why you might want to use one over the other, but you can see the results of using one compared to the other. I have a question in the back.
I'm on distance. What exactly is distance? And, I mean, I guess it would differ depending on.
You guys are just great. You're leading me right through my talk, which is fantastic. So we're talking about this distance metric, or these distance measures, and I'm not telling you what they are. So let's do this one first. Correlation. There's correlation. Okay, but I'm going to treat correlation as a black box. It's a measure of how much spread and how linearly related 2 vectors are. And then the distance between 2 samples or 2 genes in the case of samples in 20-dimensional space, or of genes in 22,000 dimensional space, is just 1 minus the correlation coefficient. Okay? everyone here has probably seen an example where correlation can lead you pretty far astray. So you might have, for example, a situation where you have a ball, so no correlation in say, two-dimensional space. You have a ball or a sphere of points, and you have a point way out here. What's the correlation? It's really high typically, right? Because this point drives your correlation up. If we use a distance metric that's based on correlation, it's going to be sensitive to those kinds of things. Sometimes we're not going to worry, most of the time we're not going to worry about it because our data sets are typically pretty large and we don't have a lot of situations where we have outliers. But in terms of thinking about your own data, if you do have situations where, for example, your measurement technology is prone to outliers, you might want to look at the underlying data after you receive a clustering result back where you use correlation to see if it's been driven by outliers. Then there's some other potential methods. Euclidean distance, I think I'm not going to spend much time talking about it. Manhattan distance, which is basically if you have 2 points in space, Euclidean distance is this distance. Manhattan distance is up and over distance. The Minkowski distance is a generalization of these two. So if we replace m here by 2, then we have Euclidean distance. If we replace m here by 1, then we have Manhattan distance. And we can change, we can use Minkowski distance to weight things a little bit differently.
So I think for the Minkowski distance, you need absolute value.
Yeah, you're right. There should be an absolute value here. Okay. I will tell you that of everything that I'm showing you here and dozens of other distance metrics, the ones that are typically used in gene expression data are Euclidean distance and correlation.
Sir.
But, yeah, go ahead.
The Euclidean distance also really affected by outliers, or how it's calculated.
It can be, yes. So this brings me to the last point here. The choice of your distance metric can and really should be driven by your underlying data. So if you apply, for example, correlation to binary data, you might actually get something that you don't expect. You can't apply correlation to categorical data, not straight out anyway. Outliers we talked a little bit about, and there are many other distance metrics that are designed for specific kinds of data. So although these things often work in practice, you might want to think about whether there's a better distance metric for your type of data. But again, the distance metric and the linkage are separate from each other. Once you have a matrix of distances, you can do hierarchical clustering. So, yeah, Naomi's got it.
So the other thing to think about is that the distance metric should also be driven by what's important to measure. Okay? So for example, correlation measures whether genes are going up and down together, but not whether one is much higher than the other.
Naomi.
Oh, sorry.
That's okay. No, you should explain it.
Sorry, I thought you were finished.
I was almost there.
I didn't want to not say that.
So, so when you guys look at this, this is Naomi's point. This is probably the most important point. When you have your data, you want to think about, as Naomi said, what's important to you. These two genes at the bottom here are very close to each other in some sense, right? These two genes are not very close to each other, but they're perfectly correlated with each other. So in correlation space, in correlation distance, what's the distance here between this bottom gene and that top gene? What's different? The distance is 0, right? They're identical. The distance here in Euclidean distance is near 0. The distance here in Euclidean space or Euclidean distance is very far apart. So again, well, as Naomi pointed out, it's really important to think about what you want to measure and how your distance metric is going to impact what you get for your clustering results. Okay.
So can I clarify?
Yeah.
Usually we're interested more in correlations, even if they're very far apart. You want to know what a gene, especially in microarrays, because the intensity is meaningless, right? Because one gene might be, the probe might not be as good as
So
why do we use Euclidean distance?
So in gene space, we might be interested more in correlation. So the question is really in gene expression, we really can't compare the absolute values of gene expression. Everything that we get back from a microarray, comparing one gene to the next, is relative expression. But in sample space, if we want to compare samples now, we might actually be interested in Euclidean space, or in Euclidean distance, because let's take, for example, a liver versus a developed red blood cell. So liver cells have to do many, many things. They have lots of genes turned on. A brain might be another example. Lots of genes need to be turned on because the liver does all kinds of stuff. A red blood cell, what's it got to do? Essentially nothing. It floats around. So there's not a lot of gene expression in a red blood cell. But there might be, in sort of absolute terms, a lot of gene expression in liver. So if we look at Euclidean distance, we could see that actually these samples are quite far apart because the gene expression in the liver is actually much higher for many genes than for red blood cells. That might not be the right thing for you, but that's the kind of thinking that people sometimes use. I agree, though, in gene expression, we're typically talking about correlation, or 1- correlation.
All of these distance metrics depart from the idea that they're pairwise comparisons where one thing is put in the x-axis and the other one is put in the y-axis. And then you kind of summarize all the comparisons?
These things are all done on vectors. So this is done over all the points in the vector. So we think of these things in 2 dimensions, right? There's an x and a y, but in reality, or in our data, we have many, many dimensional data. So these vectors, these points in space, now are in a many-dimensional space, and we're measuring distance in this many-dimensional space. Okay, you can't visualize it, but that's what's happening, and we'll get back to that because there are ways to take this many-dimensional space and reduce it into something that we can understand in 2 dimensions. Okay? So just keep your question in mind. But these distance metrics, though, do work they're working in multi-dimensional space on pairs of genes or samples.
Always pairs, right?
Always pairs. Always pairs. Yes, always pairs. Okay, so I have to put a little bit of our code in. I created a matrix. I think this should look relatively familiar. I took 10,000 random normal deviates, and I converted that into a matrix with 20 columns. So this is a data set with, say, 20 samples and 500 genes. And then to make it interesting, because this would be otherwise random data, right? I added 1. So since all of these have 1 standard deviation, I added 1 to, if this is an Excel spreadsheet, I added 1 to this little chunk of the data. Okay? So I artificially created a cluster. And then we're going to look at what's going on here. I use the hclust function, dist, for computing distance. The dist function has all of the distances that I just talked about, plus several others. And then to do the correlation, I used 1 minus the correlation, and then because hclust actually needs a distance object, you have to convert it to distance using as dist. So this is a coercion. Right? So sorry they're a little blurry, but what's going on here is that I did the clustering here using distance now, Euclidean distance, and I made dendrograms using complete linkage, single linkage, average linkage, and something called ward linkage. We didn't talk about that, but I'll talk about it in a second. Notice the differences though. So start with single linkage. How would you guys kind of describe that clustering? It's kind of flat, right? It's kind of flat clustering. There's not a lot of distinction between different clusters. So flat, sort of low clusters is what single linkage tends to produce. As compared to complete linkage, which creates very relatively tight clusters. Some people, if we could look at this in multidimensional space, these would be spherical clusters. They're tight spheres. Average linkage, you can see it's actually a little bit in between. And ward linkage creates the tightest clusters of all, right? It creates clusters that have very long branch lengths relative to the clusters. So 2 things to notice here. What happens when we cluster data? This is a really stupid question. If we cluster data, what do we get back? Clusters. We cluster data, we get clusters. Okay? That sounds trivial, but people still come to me and say, look, I got clusters. What do these mean? I have no idea. So clustering, it does. It produces clusters. And the types of clusters it produces, we can define, right? If somebody writes a paper and they don't tell you that they used ward linkage, this happens all the time. Naomi's probably seen it too. These things look like great clusters, right? But look at single linkage. Would you guys pull out 2 clusters from the single linkage result? I might not. I would have a hard time doing that. So without knowing some of the details here about the distance metric and the type of linkage, which now you guys can interpret, you can't interpret just the sheer result of a cluster, let alone the biological result. Yes?
Where does the height mean?
Height here is the distance between the clusters. Okay. Oh, sorry.
Are you going to explain what exactly does the word? Because the word option for the English.
Oh, I'm sorry. Ward, yes. So what ward does, rather than just measuring simple distance, it looks at, for each new cluster that it would create when it joins clusters, it chooses the joined cluster that's going to reduce, or that's going to reduce the variance the most in that cluster. Am I right about that? I think so. Yeah. So it basically reduces the sum of squares within that cluster. Okay. And what that ends up doing in practice looks very similar often to complete linkage. Okay. So it makes really, in multidimensional space, it makes very tight spherical clusters. Okay. That can be useful, but it can lead you astray as well. Oops.
What are the specific examples where you'd rather use single versus complete?
That's a question for you to answer. I would say, so maybe Naomi has experience where one is better than another, but the reason I'm showing you all this all started with the same data that was designed this way, and you can see, you can draw different conclusions from it, okay? I have word slides later, but I'll say it now. Again, you cluster, you get clusters. These clusters are not hypothesis tests. We didn't do any hypothesis testing here. We can't assign a p-value to these things. So what we have to do is to treat these as hypothesis generating tools, right? That generates one set of hypotheses. This generates actually a slightly different set. This generates a different set. This generates a different set. We actually know what real thing is going on. There should be, ideally, there should be 2 branches with 10 samples in each. Because that's the way I designed it. That's not actually true for any of these, right? So we know it's wrong, and it's a question of how wrong is it. Maybe this one isn't that wrong. Maybe this one isn't that wrong. But until you can attach some biological information, you go back to my circles again, until you take another piece from another circle and attach it to this, it's very hard to come up with whether or not one is better than another. So that's not an answer, but it's the practical answer. So, Naomi, you have anything to add about when you get back clusters, how to evaluate one over the other?
Well, basically, I put the class labels on instead of the sample labels.
Yeah.
So this is a little hard to evaluate because it's hard to remember. Well, it's hard to read the labels.
Yeah.
But you know, I would evaluate it with respect to what, in this case, we know the truth to be. Right. But I totally agree. It's an exploratory technique, a visualization technique, and it sort of goes along with my first lecture about looking at the data. This is a really powerful way of looking at the data, and we use it for outlier detection. We use it for to look for batch effects, right? If all your batches of arrays cluster together instead of treatment, the normal versus tumor samples, then probably you have a strong batch effect. But it's black magic. It's not mathematics or science.
Right. It's hypothesis generated. So you can take your hypothesis from this, but you really need to do what Naomi suggests. You have to follow it up with something, some other piece of information that allows you to assign significance to these things, biological significance. This is the same thing with correlation metric instead. Yes?
Is it common for people to try different correlation metrics, different linkage methods,
it's common to do that, and you guys, if you play with this, you'll find it's really easy to do. That has upsides and downsides. The upside is that you can try lots of different things. The downside is that often with biological data, you get different hypotheses based on what you try. And this comes back to the cluster stability piece. And each of these we can evaluate in terms of how well the clustering algorithm did, how well did it reduce the intra-cluster distances and maximize the inter-cluster distances. We can do that with any of these, and we can assign a number. But in terms of what they mean biologically, we can't easily do that. Yeah.
So one thing to remember, like one way to evaluate these, if you've done phylogeny, you should probably bootstrap the phylogeny. We can bootstrap this, but with the same distance metric and the same clustering method. Often these trees are reasonably stable. Well, I shouldn't say often, because often they're not, but they can be. But they're still driven by what you decided. And the example I would give is let's cluster people in this class. We cluster by gender, and we get a very good clustering, and it's a very valid clustering. Or we could cluster by what country you're looking at. We get a really good cluster, and it would be a very valid cluster, but the similarity measure is different, right? So we're going to get a different cluster. So clustering is black magic.
For me, it's a very basic question that you want to address by clustering at a very early stage of the analysis. For example, you have disease and non-disease samples. You want to cluster them.
Yeah.
And you want, somehow although sometimes the condition is not very well known. So you don't know exactly how much it should affect the expression. Right. So of course, if you do something like this, you try different distances, you try different clusterings, you might be able to find the clustering and the distance that really separates cases and controls.
But you might not.
Well, but also the question for me is, okay, fine. In this way I can separate very well, but do I conclude that the experiment went all right and there is actually a lot of differential expressions between put them? Or should I rely on another one, on another system and say, perhaps this experiment failed, or perhaps the correlation between gene expression and disease state is not the one I expect?
Let me go just a little bit further, and then because I have a couple slides that'll help, and then I have a little story to tell you.
That's sort of a related question, just to give two examples. Like, you're trying to sort of do data exploration. Say you have tumor versus normal, and you want to see if the experiment works. I mean, you would expect that they would cluster separately because the tumor was normal, right? Hopefully. But let's say you did an experiment where you have a knockdown, right, of a gene, and you have 30,000 genes in your RNA sequence. You wouldn't necessarily, and there's only a couple hundred genes that change, you wouldn't necessarily expect to see clusters.
So this is the point, yes. So here I'm doing, taking it to taking the next step. The data are in the same order as I gave them here. So if you guys, what's going on here is that we've clustered the samples. They're not in the same order, sorry. We've clustered the samples, we've clustered the genes, okay? And then we've laid the gene expression data out so that samples that are near each other are near each other in this heat map. Two things to note. The ordering of these samples and genes is arbitrary. The clustering tells us how to put two things together, but at any point, at any join, we can validly flip it, right? So the clustering that we get here, this is done in R, is supposed to allow us to see features in the data because of the way they did the ordering. But when I often get this too, these two samples and these two samples are next to each other. For example, these two and these two are next to each other. Look at the branches. Are they actually next to each other? No, they're not. So people will say, but they're right next to each other. They look like each other. They're next to each other in the heat map. No, they're not. The clustering tells us something different. So that's the first thing. And the second thing is that if you look at the data here, with clustering, if you squint, you can begin to see the features that are important up here. Remember, this was 20% of the genes. Okay? So if you have tumor versus normal, for example, typically between a tumor and a normal, across many different data sets, we have literally thousands of genes differentially expressed. Many tens of percent, sometimes even 50% of the genes are differentially expressed. So we might expect to see the tumors and normals being separated. If we take all the genes, though, in the case of a knockdown experiment, where your knockdown isn't maybe as efficient as you'd like, and there's biological variation, maybe there are just 200 genes that are truly differentially expressed, 200 out of 20,000. Your distance metric is potentially not sensitive enough to notice that those are different. Your clustering won't show clusters based on your experiment. Does that mean the experiment failed? No. Does it mean it's potentially harder to find differences? Yes. So hierarchical clustering is an exploratory data technique, but it doesn't answer the question as to whether an experiment worked or failed. Yeah.
If you take the top 30% most
Yeah, so this is a really good question. These data that I generated are all equivalent in terms of their variability, right? Because I generated random normal data, random normal variance. All these data have mean 0, standard deviation 1. In biology, that's not what we have. We have, everybody knows what housekeeping genes are, right? We have housekeeping genes that are the same across every sample. Do those help us cluster data? Because there's no information in them about the differences between the samples. So the point here is that we can use variability, we can use variance to reduce the number of genes and also get at the genes that carry the most information. So that's actually a very typical thing to want to do and to actually have to do in practice, is to take the number of genes and reduce it by choosing the top 20% or 10% most variable genes. It's valid because we didn't tell the computer which genes to choose based on classes, right? We didn't tell it to choose the genes that were differentially expressed between the two groups. We just told it, choose the genes based on their variability. The ones that are most variable carry the most information. Therefore, we want to use those in our clustering. This comes back to cluster stability, though. If I choose the top 5% of the most variable, the top 10% of the most variable, the top 20%, I'm potentially going to get different clusters. And as I move down the list, if I get the same clustering, that's maybe a little, that has one biological interpretation. But if I get different clusterings at every level, then then I'm back in the situation where I have a very small signal that I've got to tease out. So my little vignette is an interesting one that I used to teach a course in Aspen to MDs who wanted to be doing gene expression data, looking at gene expression data themselves, and we would give them a data set, and we started with this process that we're talking about is unsupervised clustering. We're not telling the computer what the classes are. We gave them this data set and asked them to look at the unsupervised clusters and define what two different types of cancer were in the data set. And they were easily able to see that they were AML and ALL by looking at the genes that are up or down regulated in the different groups. But there were 3 samples that were clearly AML, but they were different in some way. They looked like AML, they clustered with AML, but when you looked at them in the heat map, they looked a little bit different. They had a stronger similarity than the others. And so we always ask the students, so what's going on? And they always come up with, they're all oncologists, they always come up with the interpretation that, well, maybe AML has some subgroups, or maybe these patients are going to relapse later. They could come up with lots of biological interpretations for that cluster, right? So those are all great, but it turns out that the interpretation was that those arrays were run in the summer, and the dye that was used to run the arrays bleaches instantly in ozone. We're in Washington, D.C. Guess what? The arrays run in the summer were bleached. The dye on the array was bleached. So we still had some signal, but the reason the arrays looked different was totally technical. So again, this is that question of when you get your new data set, what do you do with it? And you make your clusters, how do you interpret them? And I guess the take-home message is don't interpret them too much. And you definitely need, before you jump to any conclusion biologically, even if the tumors and the normals do cluster together, you need to think first, what's the technical variation that led to this? And if you can eliminate it all, if you can sort of take out all the covariates that you think might be important, and you still have biology, then your hypothesis might be true. Okay. So here, all I did was to do a supervised clustering. I took the first 100 genes and did a clustering, and now you can begin to see first that there are 2 clusters, and second that there is some, it's subtle difference in gene expression measures. Sometimes, and this still happens, sometimes people will use a clustering method like this to say, look, I see 2 clusters, but they didn't tell you that the way that they got to these genes was to do a t-test first. So this is a perfectly expected finding. So to use this as sort of evidence that your hypothesis is true doesn't add anything to the fact that you did a t-test on lots of differentially expressed genes. Okay? Yes?
I understand what you're saying, but do you think it's still acceptable to do that if you're just trying to visualize?
This is a perfectly valid visualization technique, but if the genes are not chosen based on something that's unsupervised, something that doesn't have to do with the classes, then you certainly wouldn't want to hold this up as proof that there are 2 distinct subclasses. So we're thinking about this sort of after the fact, but I've also seen situations where people say, okay, I did my hierarchical clustering, and by one hierarchical clustering method, I found that there are 2 groups. Then I told the computer, based on these 2 groups that I don't know what they mean biologically, find me the genes that are different between these 2 groups. Okay. Then they take those genes and they make a heat map out of them and say, look, there are 2 groups in the data. Well, there are 2 groups in the data because I asked the computer to find the differences between the 2 groups that could be arbitrary. You guys talked about false discovery rate yesterday. There are plenty of false discoveries, right? We can use the false discoveries to prove our point, but that doesn't make it true. So the point is, when you're reading papers or when you're thinking about your own data, you want to think about what went into the clustering. So just going to go through some other types of clustering very quickly. K-means clustering. The input object is a number of points in multidimensional space now, and a number k. K meaning being the number of clusters. Of course, k has to be less than the number of samples, and it's typically actually much, much, much smaller. We don't often know what k is to begin with, but we could hypothesize if we had a tumor in normal that k is 2, whether we have a knockdown experiment that k is 2. We randomly place k points into the space represented by the objects that are representing the objects that are being clustered. These points represent the initial centroids. Centroids here are basically just a measure. We're going to use the concept of centroid to measure how close, or the closeness of points in space are. We're going to assign each object to the group that has the closest centroid. The centroid is now chosen randomly, has been chosen randomly. When all the objects have been assigned, we're going to recalculate the positions of the centroids. So we're going to take all the points that have been assigned to that centroid. We're going to calculate what the new centroid, what the new center of that cluster is. We're going to drop a point there. Then we're going to go back to step 2. We're going to assign all the groups, or all the objects, to the group that has the closest centroid. We're going to keep going until we get centroids that really don't change. What's this look like in practice? We're going to arbitrarily choose these 2 red dots as the initial cluster centers. We're going to assign the objects to the closest cluster, or the closest centroid. Okay. We're going to update the cluster centroids. We're going to put these points in the center of the cluster. We're going to reassign the points. So these changed colors. We're going to update the cluster centroids again. And then we're going to repeat. We keep doing this until, like I said, the centroids really don't move. Okay? The initial clusters, if we choose a set of initial clusters, then this is deterministic. One problem with k-means is that we can get trapped into local optima. That is, the centroids might not change because we found a really good solution, but maybe not the best solution. So we choose different centers for the next run, different initial centers for the next run, we might get a different answer. Okay? How to pick the initial clusters? Well, we could, for example, run hierarchical clustering and split the clusters. Let's say we have k equals 2. We split them at the top branch and use those as the initial clusters, or as the initial centroids, the initial clusters. And we could also randomly start many different times with different initial centers. Because this is using essentially a multidimensional mean, we know the mean is sensitive to outliers, right? Yeah. So it's not really tolerant to outliers or noise. Here's an example. This point way up here, it's not going to easily fit into any cluster. And it may actually drive, based on our initial guesses of where the clusters are, it may drive how our final result looks. Partitioning around medioids is, you can think of as very similar to k-means clustering. It's essentially the same process, but now instead of choosing these centroids, these averages, we just pick a point. The point's in the data set. And because it's a single point, we don't have the sensitivity to outliers problem. You can think of this as kind of like, almost like medians. Except in this case, we're not using a median, we're actually using an individual point. So it's it's more robust to outliers is the point of that. So coming back to this question about points in many dimensional space and how to think about distance metrics, distance measures. Well, in the space of samples, we have, in this case, 20 points in 500 dimensional space. Okay, so each of you, I'm sure, is picturing this right now. What if we had a way of taking the distance in 20-dimensional space and keeping it the same, or at least the relative distance in 500-dimensional space, but reducing it to 2 dimensions? Well, that's what multidimensional scaling does. The relative distances between all the points here are the same, or roughly the same, as in 500-dimensional space, but we've collapsed them onto 2 dimensions. Okay? So this isn't really a clustering approach necessarily. It could be made into one, but it is a dimensionality reduction technique. So, and these are going to be related, right? So this is the samples. These are the 20 points, or sorry, these are the 10 points that were these 10 samples, and the black were these 10 samples. Mm-hmm. Yeah, question?
Is there a way to evaluate when you are reducing the dimensionality? So to speak, like, if you start from 20 dimensions and you squeeze it down to 2, how much are you leaving behind? And how sure are you that you are having a realistic representation?
Let's see. Let's reduce the dimensionality of the data while conserving relative distances. So you could measure the distances in 500-dimensional space, measure them in 2-dimensional space, and see, are they proportional to each other for all the samples? That would be one way of doing it. And in fact, multidimensional scaling is supposed to do just that. It's supposed to take these distances, this distance matrix, and basically conserve it, but now show us in 2 dimensions. So, does that make sense?
Yeah, it makes sense. Like, viewing is the typical example of the points in the projection.
It's not a projection. It's not exactly a projection. It's just sheerly conserving the distance relationships between these points in multidimensional space.
So it should always be quite good.
It should be, yes, because that's what multidimensional scaling is trying to do. Yes. This is with samples. We can do the same thing with genes, right? And again, I've colored in red the genes that are in this top 100. And you can see that if we tried to categorize or classify these genes based on our gene expression measures, we would have a hard time doing that. I mean, we could certainly draw a line here, and we maybe are 80% or 85% classification strength versus classification ability, but not 100%. Whereas it's a little bit different for the samples. Okay. Multidimensional scaling, just like hierarchical clustering, is not a hypothesis testing approach. It's a visualization strategy. So it's just another way of looking at data. You can do this in 3 dimensions as well, or if you can think in 4 dimensions, you can do it in 4 dimensions. It works just from a computational point of view, works fine. Yes?
Is it the same as principal component analysis?
It's not the same as principal component analysis. And so this does exactly this. Principal components are a little bit different. Well, in practice, they may give you the pictures that you get from principal component analysis and multidimensional scaling often look similar. But what principal component analysis is asking is to find in multidimensional space the vector, the first principal component is going to be the vector in multidimensional space that captures the most variability. So in terms of our scatter plots, that's going to be the vector in our 2-dimensional space that captures as much variability as possible. So it's going to be, if the scatter plot goes this way, the principal component will go this way. Okay? The second principal component then is orthogonal in multidimensional space and captures the second largest vector of variation that's orthogonal to the first, and so on and so forth. And principal components then can be used for a couple of different things. You can use them to look at the features. For example, if you're looking at principal components for samples, you can look at the features of the vector to ask which genes are contributing the most to the first principal component, the most variance in the first principal component. And you'll find that the genes that are most variable are going to be the ones that are in there. And the housekeeping genes are going to be contributing to the 17th or 20th principal component. So that's one way that you can use it. You can also use principal components to ask questions about samples in that space. There are other things like principal components that you might hear about more often these days. Things like non-negative matrix factorization, or I can't think of the other one. The issue with principal components is that if you think about genes being, if you think about the first principal component and the loadings, the weights for genes in the first principal component, some of them could be negative, right? What does that actually mean? With non-negative matrix factorization, you can think of it as kind of like principal components, except the weights, the loadings are restricted to be positive. And what that means is that if you have essentially the same kind of first principal component, but with non-negative matrix factorization, you can look at the genes, and the genes that show a signal that are positive are actually contributing. They're sort of positively responsible for the variation in that principal component. So you can use it. You can use that as like with k-means or clustering in general. You can use it as a class discovery method for both genes and samples. I wasn't going to get into it, but we can certainly play with it in a lab, or we can talk more. Does that answer your question roughly anyway? Okay.
So is PCA a type of clustering, or does it fall under that?
No, it can be. I would say I would put it sort of in the category of dimensionality reduction. Naomi looks like she's got something to say.
Well, it turns out that PCA is closely related to k-means clustering. And it's also, I would think of it more as a type of multidimensional scaling. which then might reveal the clusters.
It's certainly from a practical point of view, it's a dimensionality reduction technique. But because of the way the principal components are derived, you can use not just the locations of either samples or genes, but you can use the loadings of the principal components to tell you something about what goes into it. Here, with multidimensional scaling, it's a black box. We just know that the distances remain the same, relatively. Yes?
Do you always reduce to 2 dimensions, or does it make sense to reduce to, say, 3 dimensions or something else that we can handle?
Sure. I can't do 3 dimensions in a PowerPoint, but R can do 3 dimensions and there's a little package called RGL that allows you to, like, you load your 3-dimensional object and you can use your mouse to spin and zoom. It's really cool. But yes, and as a matter of fact, if this were in 3 dimensions, so this is only 2 dimensions, it's possible that in 3 dimensions we could find a plane that actually separates these genes perfectly. We can't see it here, but it's possible to do that. And actually, in many dimensional space, now if we don't have to reduce this, if we allow the computer to do the work, sometimes we'll use classification techniques that do exactly that. They try to find planes that divide the samples. But now the plane is not in 2-dimensional space, or not in 3-dimensional space, but is in 1-dimensional space.
What's the name of the R package you mentioned?
Sorry?
The name of the R package you mentioned that allows you to create
RGL.
RGL.
Yeah. Okay. We talked about this a fair bit already, but I'll just go through it in a teeny bit more detail. Feature selection and clustering. We talked about 2 sort of extremes of feature selection. One is totally unsupervised. We use something like variation to select genes for clustering. The other is totally supervised. We do a t-test first and then use the genes from the t-test to do visualization. But we could also choose, for example, particular pathways. In cancer biology, people ask me people who are working in drug development ask me all the time to make hierarchical clusters of all the receptor tyrosine kinases. So I choose all the receptor tyrosine kinases, all 600 of them, and I hand them back a heat map of their samples and their genes. And they use that to help them figure out where to go next in terms of drug development, for example. Particular gene functional categories, transcription factors. You might want to see, are there groups or there batches of transcription factors, HOX genes that are coordinately regulated. You can choose them out of your gene set, use hierarchical clustering as a very quick, hierarchical clustering here I mean heat maps as a quick data visualization technique, gene cellular localization, etc. And again, just to beat a dead horse, if genes for clustering are chosen based on differences between groups, then we call it supervised.
Sorry, just to be clear.
That's okay.
Differences between groups. Did you do a statistical test? Because it's saying just particular pathways could be differences between groups. Is that okay?
By differences between groups here, I'm talking groups of samples. Differences between groups here, I'm talking groups of genes. Okay, so I'm using my sort of human genomics view of the world, that I'm often interested in how the samples relate to the underlying gene expression. So if I want to know how my samples are clustering and what the underlying gene expression looks like for receptor tyrosine kinases, I can validly choose those genes because I didn't tell the computer that there are differences between my samples. I said, just give me this category of genes. There may be no differential expression in those genes. Okay?
So
Supervised means that we did the t-test, or we did the ANOVA, or we did a ratio of 2 groups, or something else. We did something that either we ourselves used the sample identifiers or the sample information, or we told the computer the sample information. Okay. Yes?
So selecting the highly variant ones would be supervised?
So The rule of thumb is if you select the genes based on the sample labels, then that would be supervised. So if you select the genes based on some category of genes, but you don't use the sample labels, that's unsupervised. Okay. Does that make sense? Okay. Validity of clusters. Again, now we're getting close to summary. Given some data, any clustering algorithm generates clusters. So we need to make sure that the clustering results are valid and meaningful. We can measure the validity of clustering based on optimality of the clusters, minimizing the within group distances, maximizing the between group distances, parsimony. Parsimony here means if we're talking about hierarchical clustering, probably 2 clusters is a better clustering than 27. Maybe not, but probably. So oftentimes we're going to choose the lowest number of clusters because that's the most parsimonious answer. Verification of the biological meaning of the clusters it's absolutely, you have to do it. I mean, if your experiment involves, if your data analysis plan involves generating some data and doing hierarchical clustering to see what I find, you really want to think about whether or not, you want to think about the next step. I do my hierarchical clustering, now what do I do? How am I going to verify this? If you're working in, for example, human samples, you might want to think about, I'm going to need another patient population. If you're working in the biology lab, you need to think about, do I have enough of this cell line to be able to do the biology to show that the genes that I find that are supposed to be responsible for my phenotype are actually doing something. So that kind of thinking, this is going back to Naomi's experimental design talk. It's not just a question of coming up with the analysis. It's a question of discovering biology, and that's going to potentially mean following up on the hypotheses that are generated. Note that the ordering of branches is arbitrary. That's important. And clustering, hypothesis generating. It's not hypothesis testing. I don't know how many times I've said that, sorry. All right, so the slides are online. This is just, if you guys want to follow up at some point, Thomas Skirke's clustering tutorial, that's a clickable hyperlink, and he's got a bunch of clustering examples there, including some relatively newer clustering algorithms. The cluster package has many different clustering algorithms. Some of them hierarchical, some of them are agglomerative, actually, are divisive hierarchical clustering. Other types of clustering as well. You can just type example and then the name of the clustering algorithm, and it will run an example for you. And if you really want to be overwhelmed, do help.search cluster, and you'll probably find that there are dozens or hundreds of R functions that have the word cluster associated with them. So, all right, I'll stop there. Any questions? All right. Thanks for all the questions during. That's great. So just a quick detail thing. If you guys wouldn't mind, so it's 11:05. If you wouldn't mind sort of on your way out to an early lunch, if you could run over to the computer lab on the Sakai website. On the Sakai website, underneath Mark's talk this afternoon, there's a setup for BioC annotation lab. You just click that, it's going to give you this little text file. And just cut and paste that into an RStudio session. That's going to do the download and installation of the packages for Mark's lab, hopefully. And then, because some of them are a little bit big, it'll run over lunch, and then hopefully it's done when you guys get back. All right? Thanks.